Hi, Bio 30. I'm going to provide material from slides 39 up to slide 73 today. Okay, we are, I'm going to finish up the first section of genetics. You'll have all the material you need in order to be able to do your mitosis, meiosis, and sexual and asexual reproduction unit tests after this. And then I'm going to pause on the lectures for a few days so you can get caught up with your mitosis and meiosis quizzes and your meiosis comic strips and do your unit test. Okay, so I ended up talking about meiosis the other day. We talked about karyotypes as well before that, looking at uneven chromosome splitting, right? Things like Turner syndrome or Down syndrome, where there's extra chromosomes or chromosomes missing in certain homologous pairs. So when that happens, that's called non-disjunction. So if we apply this to meiosis, you know when chromosomes split. So chromosomes split. Obviously, the homologous pairs split in meiosis in anaphase 1, and then the sister chromatids split in anaphase 2. So this is where non-disjunction happens. If I were to put this on the board, right, so you have your diploid germ cell, and then it replicates its DNA. After S phase, and then you're going to have your homologous pairs with sister chromatids attached. Now remember, in meiosis 1, homologous pairs typically split. Okay, and then in meiosis 2, the sister chromatids split. All right, now during gamete formation, if non-disjunction occurs, you might have issues like this. So let's say we have our homologous pair here, and then let's say we have uneven chromosome splitting that could occur. So this could happen, so maybe one sister chromatid gets split and goes here, and then the other sister chromatid is left. So there's a full homologous chromosome with a sister chromatid, and then the other homologous chromosome is split. So there's 3 pieces of genetic information after meiosis 1 in this cell, and then there's only going to be 1 piece. So remember, right, there's 4 pieces of genetic information for each homologous split. And then in meiosis 2, what could happen is, right, you'd end up with a homologous chromosome here, and then the other homologous chromosome with its sister chromatid there. And then on this chromosome, one cell might not even have a chromosome, right? Because there's only 1 piece of genetic information. So let's say this gamete fertilizes another gamete, and there's an extra chromosome. So this extra piece of genetic information would act as an extra chromosome, and that could lead to an uneven, an extra chromosome on like chromosome pair 21, which would lead to, Down syndrome, or Klinefelter syndrome, or something else that would have 47 chromosomes rather than 46. This would be a normal gamete. This would be a normal gamete. And then this gamete here, it could be either egg or sperm, right? This actually wouldn't have a chromosome. So if this fertilizes another gamete, there'll be not enough chromosomes present, right? So you have 45 chromosomes. So this is how non-disjunction occurs. So this uneven chromosome splitting, it happens at anaphase 1 and anaphase 2 of meiosis. It happens more commonly in meiosis 2, where these cells divide. But obviously it's more harmful in meiosis 1. So you can see after meiosis 1, you've messed up both parts of the division. In meiosis II, you're only going to mess up one side. Okay. So, that's in my notes. Right, so non-disjunction produces gametes that have either too few or too many chromosomes. So if there's too few, we would call that monosomy. So one chromosome is lost due to non-disjunction. We already talked about that with Turner syndrome. The other common type of non-disjunction is trisomy, where there's an extra chromosome when the sister chromatid doesn't split when it should. That's what causes Down syndrome. That's what causes Klinefelter syndrome. That's what causes other genetic non-disjunction disorders like metabolic triple X syndrome, so on and so forth. Okay. Right. So this is these are just some diagrams of individuals that have Turner syndrome and Down syndrome. Right. Down syndrome is quite common. Right. We all probably know someone with Down syndrome. Turner syndrome is less common, but they present as female. 45 chromosomes, only one X, and there's lots of developmental and intellectual abnormalities there. Okay. We'll talk about how gametes form. So basically this is looking at meiosis in an applied fashion. So the formation of sperm is called spermatogenesis, and the formation of eggs is called oogenesis. They happen with meiosis, obviously. But I'm gonna show you some different comparisons in your textbook. I'm just gonna go to page 563 in McGraw-Hill. Page 569 gives you kind of a timeline of oogenesis and spermatogenesis. Okay. I'm going to draw these out and show you the differences, and then I'm going to draw a little Venn diagram comparing the two. So spermatogenesis doesn't happen until puberty in the male, all right? That is different from oogenesis, because oogenesis actually starts in utero with females and then kind of gets frozen in time for a little bit until puberty begins. So just a reminder, with spermatogenesis, this happens in the seminiferous tubules of the testes. You have the Sertoli cells that nourish the sperm. And then interstitial cells will be kind of on the outside borders of the seminiferous tubules. And what they will do is produce testosterone, which aids in sperm production. Okay, so what do we start with? We start with a germline cell. It actually starts with a mitotic division. Okay, so there's a mitotic division, and that keeps, so one cell will go through meiosis, and the other daughter cell stays in the germline. So both these processes actually start with mitosis to keep the same amount of germline cells. Okay, so one ends up here. We get S phase of interphase happening. Okay, so S phase of interphase happens. And then we have a cell that so a cell that has replicated its DNA so this is called, once that has happened, that's called a primary spermatocyte. So your germline cell is called the spermatogonium. Okay. And then once S phase has happened, you have the primary spermatocyte. Okay. From there, you get meiosis 1. And then you get the homologous pairs separating. These are called secondary spermatocytes. Okay, and then you'll get your second meiotic division. We've already talked about the stages of meiosis. And you'll end up with your immature gametes. So these guys, okay, once the homologous pairs have split and you just have sister chromatids left and you have haploid cells, these are called spermatids. And then these end up going to the epididymis, and they get tails, right? They get flagellated, which is a fun little word. Your sperm all get flagellated. Wonderful. All right, and that is, and those are spermazoa or sperm. So this happens continuously at puberty, and in the average adolescent male. The average adolescent male makes about 300 million sperm a day. And that gradually tapers off as males mature. But there's lots of ways for males to maintain sperm count with regular exercise and maintaining testosterone levels and living a healthy lifestyle and things like that. So males typically are technically fertile for their whole life after puberty has happened. So this is a slide that just explains what I just put on paper. I'm going to leave it there. You can go over that. Sperm gain flagellums, like I said, in the epididymis. I already talked about sperm anatomy and reproduction, so you can review those notes if you want. Oogenesis happens really similarly, but there's some significant differences. So again, we're looking at meiosis in a sort of an applied fashion here. So oogenesis obviously doesn't occur in the testes. That would be weird. It occurs in the ovaries in the female. Oogenesis starts the same thing with a diploid germline cell. And rather than it being called a spermatogonium, it's called an oogonium. Okay. And it starts with mitosis again, right? So there's a mitotic division. So one cell will go through meiosis, and one cell will remain in the germline as a stem cell. Okay, then this replicates its DNA, right? And this is the S phase of interphase. So the terms are really similar. So this would be a primary oocyte once DNA has been replicated. Okay. I'm going to turn to this diagram right here. So you can see this is kind of looking at reproduction again, but this looks at ovulation, the corpus luteum, and all that stuff. So this actually, where this oocyte happens, where it's formed, this actually is formed in utero. So a female is born with all of the eggs she will ever need, because they're produced during fetal development. They just kind of get frozen in time for a bit. So they actually get arrested at prophase 1 until puberty happens. So none of the developments of meiosis occur until puberty. And even then, most of the oocytes don't form into eggs. They remain dormant. It's kind of only 1 or 2 a month, right? Based on a female's menstrual cycle. That will go through this process. There's also another big difference here, is that there's uneven cytoplasmic sharing. So when puberty starts, there will be one egg So this is a primary oocyte. And then this is a polar body. Okay, so remember the goals of the female and male reproductive systems. The goal for a male is to produce a large quantity of offspring. So there's not a lot of investment in gametes, right? It's just kind of spray them wherever you can, right? With a female, because a female actually has to give birth and do all that, right? A female's gametes are wired for quality, right? So you want high quality gametes fertilizing your eggs, right? So there's investment in a single egg during meiosis, right? So you have your homologous pairs split. There's uneven cytoplasmic sharing, and this polar body degenerates. Okay, and then meiosis. So this is meiosis 1, and then in meiosis 2. This polar body will produce 2 more polar bodies. Those are not used for fertilization. They'll generally be reabsorbed back into the ovary of the female, back into the body tissue. So you'll have 1 egg and 3 polar bodies after meiosis 2. Okay. After meiosis 1, you have secondary oocytes, not primary oocytes. And then you'll have, rather than a spermatid, you'll have an ootid. And then that'll develop into an ovum, and that'll be haploid, right? So these polar bodies typically degenerate. So a female produces 1 high-quality egg at the onset of puberty each month. Now, in some animals, these polar bodies, what they can do is they can actually fertilize the egg that's produced. And remember, with meiosis, one of the big things that meiosis does is recombination. So we swap alleles between homologous pairs. So none of these gametes are genetically identical. So there's a lot of animals that can just fertilize, they're females, and they can just fertilize their own eggs and create genetically diverse offspring. So males are not necessary to create genetically diverse offspring. In fact, males probably only exist from a biological perspective to increase genetic diversity during times of stress. All you need to do is look at the X and Y chromosome. The Y chromosome is really, really small. It's only about 14 genes. And this determines whether the erectile tissue will be male anatomy or female anatomy, because testes determining factor is in that Y chromosome. So males are accessory most of the time. I bet most females would agree with that. All right. So, oogenesis, right? So the secondary oocyte undergoes meiosis 2. This is called uneven cytoplasmic sharing or asymmetrical cytokinesis. This ensures only 1 egg is produced and nutrition for that egg is maximized. All right, so there's a bit of a phase lag in oogenesis. So you can read that slide and see my analogy. Captain America should have been female. Anyway, so let me draw a little Venn diagram here comparing spermatogenesis and oogenesis. Okay, so things that are similar between the two of them. They make haploid gametes. Right? They both make cells for reproduction. Okay, they both indicate sexual maturity. But the purpose of spermatogenesis is to produce a large quantity of gametes, and with oogenesis is to produce a few high-quality gametes. Okay, with spermatogenesis you get 4 sperm per 1 division. With females, with oogenesis, you get 1 egg and 3 polar bodies. Okay, I would also add with similarities, there's 2 divisions. There is uneven cytoplasmic, with spermatogenesis, there's even cytoplasmic sharing, or symmetrical cytokinesis. So they all get the same amount of cytoplasm. And with oogenesis, there is uneven cytoplasmic sharing. So one egg gets all the cytoplasm and the rest don't get much. With spermatogenesis, this is continuous at puberty. And there is essentially no timeline if a male remains healthy. Right, if you look at oogenesis, there's phase lags. Throughout a female's lifetime. And then there's also a timeline, right? Females go through menopause. They will not produce eggs after a certain age. And that's largely genetically based. Okay. So when we talk about fertilization, we could talk about different types of twins. Twins happen in about 1% of all births. You can have fraternal twins or identical twins. Fraternal twins is when a female produces 2 eggs and both of them get fertilized. So they're born at the same time, but they're no more similarly related than siblings. Identical twins is when a single egg splits into, right? So a single egg is fertilized, and when the zygote is formed, okay, the zygote will form into 2 blastocysts. All right. So identical twins are much rarer, right? So these are, and this is kind of old now, but right, see, they're identical twins. The Olsen twins from Family, from Full House, are fraternal twins. These are the Condro brothers that were graduated from St. Tim's about 10 years ago. They were fraternal twins. They both ran for the Dinos. When we look at division, we can look at application of non-disjunction in mitosis. So we induce non-disjunction with ultraviolet radiation and subtle genetic engineering. So we'll induce non-disjunction into plants. Sometimes this happens in plants naturally. But in order for us to make bigger and more economically appealing fruits and vegetables, we will introduce polyploidy into a lot of these plants through mutation and genetic engineering. And this is part of what makes genetically modified organisms part of it, right? The other part is that we add genes in through genetic engineering. And we'll talk about that in the last unit. But this, right, if a strawberry is, the effect of this with plants and animals, not animals as much, sorry, plants, is that, if a strawberry is diploid, for example, if I induce triploidy into a strawberry, it'll grow bigger and it'll be a more valuable economic product. So we mess around with the non-disjunction when gametes are formed in our agricultural products in order to be able to produce more valuable products. So some people are creeped out by that, but it happens all the time. Okay. So that's spermatogenesis, oogenesis, non-disjunction. We're going to talk about asexual and sexual reproductive strategies. And hopefully I'll get through this relatively quickly. All right, so I'm going to talk about different strategies between the two of these. So let's define what they are first, because it seems obvious, but it's not as obvious as you think. So asexual reproduction involves mitosis, creating an exact clone of the parent, and fertilization is not required. Okay, so for the most part. There are some examples that are a little bit diverse from that though, right? Sexual reproduction is when gametes are produced by meiosis, and the fertilization is generally between genetically distinct parental gametes, so usually male and female. And that produces distinct, genetically diverse offspring. But I've told you some examples that sway from that already. So in reality, most animals have a combination of mitosis and meiotic stages in reproductive processes. I just showed you that with spermatogenesis and oogenesis. But there's also a wide variety of different life cycles. You also have the majority of animals in organisms on Earth actually switch between asexual and sexual reproductive strategies. And we think that's weird because humans can't do that. But that is more the norm than having sexual dimorphism, meaning distinct genders. All right, so things are a lot more fluid than you think or would like to believe. So prokaryotes and eukaryotes have different forms of reproductive strategies. I'm going to talk about that now. So again, prokaryotes are organisms that don't have membrane-bound organelles. So they have to do everything with their cytoplasm. That's why they typically are single-celled, but they don't have to be. Eukaryotes are the kind of cells we have. So we have membrane-bound organelles and we have differentiation, right? So we'll have parts inside the cell, like nucleuses and mitochondria and endoplasmic reticulum and defined organelles with roles. Okay. So prokaryotic reproduction, the typical type of reproduction is a very simple form of mitosis called binary fission. There's all there is cytoplasmic DNA. But you also know, particularly viewed in Science 30, that bacteria and archaea, which are a primitive form of bacteria, and other single-celled organisms, they not only have just their cytoplasmic DNA, they have plasmid DNA as well. So prokaryotes have DNA that is replicated but have no nucleus. So mitosis doesn't occur to the same extent that we would think it does. So in good conditions, bacteria can reproduce every 20 minutes. So because they reproduce so quickly, mutation can build up relatively fast, right? So that means they can build up resistance because a mutation might be selected for, particularly if there's really massive natural selection going on in that population, like if a bacteria is being hammered with an antibiotic, resistance builds up faster. You just speed up the evolutionary process, right? So mutation can arise really quickly because doubling time is very short in these organisms. The problem is that they're all clones, right? So how do they get around that? Bacteria have plasmid DNA. So bacteria not only have, I'm going to talk about this more with the last section of the unit, but they not only have chromosomal DNA, but they have plasmid DNA. So this plasmid can be transferred between different bacterial species. Okay? So this plasmid DNA can be transferred between different bacterial species to increase genetic diversity, and that can happen really quickly. So these are two different species of bacteria. Okay, so one is E. coli and one is strep, or streptococcus. So here's what happens. So this is kind of like bacteria sex, all right? There's this stream of cytoplasm between the two bacterial cells, and this is called a pilus. And this cytoplasmic stream is what they use to exchange plasmid DNA. And again, this can increase resistance really, really quickly. So not only do they have a short generation time, they also have plasmid DNA. Okay, so that's how bacteria kind of get around being asexual. So we'll talk about asexual reproductive strategies in eukaryotes as well. So a lot of these eukaryotic organisms can reproduce sexually if necessary, but they don't always need to. So I'm going to talk about when it's advantageous to reproduce sexually and when it's advantageous to reproduce asexually. So why would an organism want to produce sexually? Well, the evolutionary reason is it increases genetic diversity. And again, I've talked about this with meiosis, right? You have gamete swapping during crossing over. You have two sets of genetic information that haven't existed before together when fertilization occurs. And you have the random assortment of alleles during gamete formation as well, right? So sexual reproduction is advantageous because environments are usually changing. So you want to have different sets of genetic characteristics to be able to handle different conditions. So you can reduce competition between siblings with genetic diversity. If they were all clones, they'd have similar strategies and similar, maybe even similar personalities. Pairing of homologous chromosomes can shield recessive harmful alleles. I'll talk about this more with Mendelian genetics. But most genetic abnormalities are things that are on recessive alleles. It's things like cystic fibrosis that you inherit. Crossing over offers opportunities to replace or repair damaged chromosomes in meiosis. So you get a genetically diverse, robust population with sexual reproduction. So you want an individual to reproduce sexually in a changing environment. And for the most part, environments are in constant flux, right? You can see the seasons happen every year. There's environmental changes. There's species moving in and out. It's a very chaotic environment. So sexual reproduction is usually advantageous as a result. But asexual is cheap and easy, right? So it's easier, it proceeds quickly, it's less energetically costly. Think about how complicated it is to find and maintain a girlfriend or a boyfriend when you're a teenager. Who wants that stress, right? So it doesn't require the presence of a second parental organism. Organisms may, and here's the other deal too, right? With asexual reproduction, these organisms might live in a stable environment. So why would you want to reproduce sexually when it's just a hassle, right? If I have like gut bacteria, like H. pylori bacteria that lives in my body, and my body goes to great lengths to maintain homeostasis, it doesn't really make sense to reproduce sexually because I need the same set of genetic characteristics to survive and thrive regardless because my environment never changes. So in a stable environment, being asexual is advantageous. Okay. As well, asexual reproduction can maximize chances of survival because the daughter organism doesn't fully separate from the parent. Okay. So if you look at the poplar groves around here in the ranch house, and poplars are pretty much the only type of deciduous tree around here. They are all clones of each other. So there was one parental organism, and then it had shoots that were cloned off of it, and they turned into another tree, and that's all a single organism. Okay. So if you stay in the same environment or move very little your entire life, it may be more beneficial to have asexual strategies. So this is funny. So when you look at the spectrum of gender, asexuality is actually a thing. And if you look at the asexual pride flag, those are actually St. Tim's colors. I don't know if you should read too much into that, but there you go. All right.